You're listening to Let's Talk Creation with Todd Wood and Paul Garner, the creation show where we learn, grow, and worship.
All right, welcome back to another episode of Let's Talk Creation with Todd and Paul. I am Todd Wood.
And I'm Paul Garner.
And we're here today on a chilly afternoon here. It's almost February when we're recording this. And we're going to do a little more natural evil in our natural evil series. So you might remember we have had a number of episodes in this series. We've talked to people about parasites, which are gross. We've talked to people about rattlesnakes, which was fascinating.
Yeah.
All the time trying to understand where the less nice parts of creation come from. How do they fit into God's good world, right? The end of creation. Week, Paul, you know this. God saw everything that he had made, and behold, it was very good. Right?
Very good.
And we all know that one. Yep. So now we have, grumpy grizzly bears, and we've got these snakes and all this good stuff. And so I say good stuff, and yet it's kind of unpleasant, right? And so we're trying to sort of figure this out. Now, there's another dimension of that which relates to our guest for today. And that dimension is the defense mechanisms, right? So it's easy to talk about the things that hurt us. That makes sense. I can understand why grizzly bears would be something you'd want to watch out for in the original creation if they were there. But on the other hand, there are also all these remarkable defenses, things like turtle shells or how fast deer can run, things like that. These are all related to coping with the sort of bad stuff in creation, the basically what we would think of as natural evil, the things that cause suffering. Okay, so that's another aspect of this whole issue. How do we incorporate all this? If the original creation was very good, and it was generally benign and absent of suffering, and if creationists are right, young age creationists are right, and there's no predation at all, there's no animals eating other animals, then why do animals need to have all this great armor, right? Why do we need armadillos? Why do we need armadillos with armor on them? And why do we need turtles with armor on them? And why do we need horns on cows and antlers on deer and so forth? What's the purpose of that? Okay, so today's guest is Dr. Joe Francis. Joe's been on this podcast before. Welcome, Joe.
Welcome. Great to be here.
Yep.
Hi, Joe. It's great to have you back.
Thank you. Good to be here.
And if you don't remember, Joe is a microbiologist, immunologist. He has a long career in Christian academics. He was a professor at Cedarville for a while, and then he moved out to the Master's College, which became the Master's University while he was there. I'm sure that's why it became the Master's University, because he's such a big shot. You can't just be a college to have, right? So and then you were dean of something for a while, dean of one of the colleges.
Yeah, the sciences at Masters, and now going close to 20 years at Liberty Online. So.
Yeah, Liberty Online. That's right. You're still active with Liberty Online?
Yeah, I've even moved up to instructional mentor, whatever that means.
Wow,
that's a good one.
Is that good? Does that just mean more work?
Oh yeah. It pays the bills.
So you've retired now from Masters.
Online with them a little bit too, but yeah. But I secretly watch over Dr. Matt McLean. So
right.
He needs watching over.
Matt is your the one who has taken up the mantle and has somehow gotten himself into a dean position there. I feel bad for him. Yeah, absolutely. Much prayer needed. Yeah, I administration is not my game. I'm happy to be just a lowly peon. But here I am running the show at Core Academy. So yeah, I don't know what I'm doing. I'm crazy. Anyway, today's topic is all about the immune system. So let me just set the stage just very briefly here. Animals today have, when we're definitely talking in the context of animals today, they have a remarkable system of cells and proteins and genes that all work together to identify intruders in your body and get rid of them. So if you've ever had, and I think the easiest way that people can imagine this, if you've ever had any sort of infection, even a a really bad pimple, you have felt the effects of your immune system. That's your immune system. Those that grossness that's in that infection, that's all a combination of bacteria and your own cells fighting off the bacteria. You get a little localized fever there because that's part of your body's response to get rid of these bacteria. And so it's one thing to think about how the world before the fall might have been absent of disease, right? And absent of those kinds of bacteria that do that nasty stuff. It's quite another thing, I think, when you sort of think, well, why then do we have such an elaborate immune system? What's it for? Mm-hmm. So that's our subject today, and you are going to walk us through. I'm thinking maybe we should have introduced this topic a little bit more, but that's okay. Let's talk about the major histocompatibility complex. And for those of you listening or watching this, the MHC, we call it MHC for short. It's a remarkable part of our immune system. It's one component of the immune system. And we're going to try to make sure that this is as understandable as we can possibly make it. Because I know your eyes just glazed over when I said major histocompatibility complex. So, Dr. Francis, can you explain to us in 5 minutes or less everything you know about the MHC? Probably not, but at least explain to us what in the world this thing is and why is it cool? Why do we care about these things?
Yeah, it's a tough subject. Unfortunately, it's put near the back of the textbook. I don't know why, because it plays a central role in the immune system. But what I'm going to do in 3 minutes is first, let's get a handle on the basics of the immune system, and then I think we'll see why the MHC is so important, so relevant. When I start out my class, I start out with two ideas that we can put together, and one idea is that the immune system, we can think of it as a recognition and response system. In other words, it has to be able to recognize something that's foreign and could hurt us, and if it has to decide if it can hurt us, so there's a response. Another way to look at the immune system is look at the innate and specific parts. And now that's a little more confusing. What do you mean the innate versus the specific? So what we're saying is it has two big branches to accomplish recognition and response. And those two big branches are the innate and the specific, or we could say adaptive system. So let's break that down. What do we mean by innate? What we mean by innate is defending ourselves from microbes that are pathogenic or potentially pathogenic is an hourly, minute by minute, even a minute by minute job. And if we want to talk right now about a creation perspective, it's God's mercy. Whenever I teach this class, within the second or third lecture, it's God's mercy that he made a system that protects us it's actually minute by minute. So why is that relevant to innate? Innate means these are the first responders. So right now we have these white blood cells making up a good portion of our blood. They're circulating. Some of them actually are going out into the tissues, and we have antibodies. And those are innate, meaning they're not thinking about anything. They're just ready to respond. They're the firemen that are right there. They're not at headquarters. where the chief fireman's going, you go here and go there, they're already on the scene. So innate means on the scene. These white blood cells, if they see anything foreign, they'll gobble it. And sometimes that's bad because maybe that splinter in your finger starts to get red and swells up. That's not so much just the infection, that's the white blood cells just dumping every chemical they can at it. And of course, our understanding of this has made millionaires. There's a German guy by the name of Bayer aspirin guy.
That's right.
And they've been millionaires for what, centuries now? Because they figured out if we can stop, if we can help the farmer's finger not hurt anymore, he can go out there and work. So, and then ibuprofen. I mean, everyone wants ibuprofen. That is anti-inflammatory. So we know about this minute by minute thing because we take our ibuprofen every other day or every week or month to stop that response. It's an overkill, but it's a good overkill. So innate means first responders. Then we have this specific system or adaptive system that says, wait a minute, you guys are doing a good job, but there's viruses that can be inside our cells that are kind of cloaked. And how the white blood cells are really just, I don't want to say they're dumb. They're just, hey, we got one job to do, kill whatever we see, but they can't see a virus inside a cell. So the specific system basically says, we want to know what's inside the cell. And this is where the MHC comes in, the specific adaptive part. So wait a minute, how does an MHC, which is a receptor on the outside of a cell, tell us something about looking inside the cell? And that's where we're at today. The MHCs are their gene, that we're talking about is genes. They're genes. Genes are a piece of DNA that code proteins. Those proteins are receptors. Every one of your body cells has MHC on it. You say, wait a minute, that sounds like a first responder. Well, it's kind of the secondary, it's kind of the link between the chief firemen and the guys on the first scene. So here's what happened. Let's talk about viruses. So, and in fact, let's back up. There's two aspects of MHC. Okay, there's MHC1 and MHC2. So in every cell of your body, you have these receptors called MHC1. And they're very good at looking for small peptides that might be foreign. Now, wait a minute. We're just talking about bacteria and viruses. So here's what happens. Inside your cell, all your cells that have MHC receptors on the surface, they're waiting for a report from inside the cell. This gets really intriguing. So what happens is, as the MHC protein is being made, the cell starts loading its receptor surface with peptides. And when they get to the surface, the T cells come along and say, there's an MHC molecule that could activate me, but he's got this little peptide that is self. Okay, so let's back up now. MHC is a receptor on the surface. It's interested in finding what's inside the cell. Okay, the MHC on every one of your cells is going to report to a T cell. Now, what's a T cell? A T cell is probably the most important chief fireman here in your system. It's looking to make sure every cell is not infected. That's a big job. So this T cell, we call it a T cell because it comes from your thymus. So, you know, you have your thyroid up here somewhere. I'm not an anatomist, and your thymus is up there somewhere. And the thymus produces these lymphocytes. Lymphocytes are another white blood cell that are chief firemen that are everywhere. So this is like you're the fireman and your chief's right there. Okay, so these T cells have a receptor that engages with the MHC receptor, and and the MHC has a groove, and it puts a little peptide. And what's a peptide? So what happens is inside every cell, as proteins get old, they're chopped up, and they're put inside the groove of the MHC molecule, and it goes to the surface. And the chief T cell, okay, which has a T cell receptor, comes over, and they engage, and it goes, okay, you're okay. You have a self protein. That's your protein. It's not foreign. And it backs off. So now a virus comes along. The virus gets inside. The phagocytes have missed it. There's no antibodies that bind to that virus because you've never seen that virus before. And that virus is going to start multiplying. So inside the cell, this chopping up procedure goes on of all the peptides. And now an MHC1 molecule goes to the surface. The T cell comes over and says, that is not you. You are infected. And it instructs another T cell called a cytotoxic T cell. Cyto means cell, toxic means toxic, right? It tells a cytotoxic T cell, come on over and join us. And that guy, that cytotoxic T cell, releases some wicked chemicals, enzymes that just destroy that cell, destroys your body cell. But you can see why. You don't want that body cell to multiply with those viruses. That's going on probably minute by minute also. Now you say, wait a minute, but viruses can overwhelm. Yes, they can, because there's a lot of them and they can multiply and so on, but the report usually goes out. So like in COVID, it wasn't the system breaking down. It was the system being too good, because one of the things that T cell can do is call more innate system over. Hey, white blood cells, come over to the lungs, come over to the lungs, come over to. And so it was inflammation that was killing us in COVID. It wasn't so much the virus taking over. It was the system being overwhelmed and bringing in too many white blood cells. So, but you see the central role of the MHC. The MHC is a reporter. If we don't have MHC, we don't report to the boss. The boss is in the dark. He can't call over help. He can't call over cytotoxic T cells. He can't call over any more white blood cells. And the other thing that joining does, that T cell can tell another lymphocyte called a B cell to make antibodies. So it's a beautiful, what an incredible system to fight infection. You got a cytotoxic T cell saying, okay, destroy this cell. Wait, there's more viruses over here. Phagocytes come over, clean it up. Oh, let's make some antibodies, and the antibodies can tag that virus and make the white blood cells see it better. So MHC is really important. That's MHC1. But here's where it gets tricky. And that seems all to make sense. What I see in immunology is a redundant backup system. I see a creator saying, you're in a fallen world and you're infected minute by minute, and I'm going to show you my mercy minute by minute. And now maybe we see a reason for this. Doesn't solve the problem where the bad thing came from. But I think what we've missed in this discussion is God's mercy. Right now, you have a phagocyte checking out, whatever you touched this morning. He's there going, he's not going to know anything happened, but I'm going to clean this up. So we see God's mercy. But let's go to MHC2. Here's where it gets a little more complicated. With MHC2, that's a set of genes that makes receptors that, guess what, has a binding group for small peptides. And again, why is it using these small peptides? Why is it chopping up the bad thing into small peptides? Well, probably because, remember, the recognition is part of the immune system. The more parts of the bad guy you can recognize, the better. The bad guy is fairly big. A virus can be fairly big, okay? And so by putting things in that groove, you can find a T cell that is specialized for that part of the virus. And you can find a T cell that's specialized for this part of the virus. So it's very, very thorough. So, well, what about MHC? So MHC, we're going to add a backup level here to this whole scenario. We just killed a virus with a cytotoxic T cell. MHC1 helped us recognize the foreign part. So what happens, though, is we have professional phagocytes. Phagocytes, again, are like those innate cells that just go surround and internalize. That's what phagocytosis is. Surround, internalize things, and kill them inside. So we have professional phagocytes called antigen-presenting cells. Okay, so what's the antigen? The antigen is anything that's foreign. The antibody binds to an antigen. Antigen is anything that's foreign. So everything we're putting in that groove are pieces of antigens. So antigen is a bigger concept. It gets chopped up and put in the groove. So now these professional antigen-presenting cells, they have both MHC1 and MHC2 on their surface. But what they do is they're kind of a backup system. They say, we didn't find anything in a cell, but we found something over here that's kind of floating around. And they put it inside their cell. They phagocytize it. They load an MHC2, And that alerts now another T cell to come over and say, hey, we got a virus that's not inside a cell, it's loose. We need more activity. So MHC2 activates, remember I told you in MHC1, it activates a cytotoxic T cell. In MHC2, it activates a helper T cell. What that helper T cell does is alert more T cells and more phagocytes to get involved. So you see the redundancy here. It's really backup. So MHC1 is on all your cells. That's being surveyed every minute, it's every day. You can have thousands of receptors, MHC receptors on each of your cells, thousands of receptors putting peptides up all day long. T cells are coming over, hey, it's a slow day, nothing going on. Everything's self. I'm just, MHC1 saying, you're good. This is just going on. So, but every once in a while, when an infection starts, this is where the antigen presenting cells say, you know what, we need more help. We got to get these helper cells. They will help us activate more phagocytes. They will help us activate more cytotoxic T cells. They will help us figure out what's going on. And also, where this whole system works too is with cancer. Because when we form cancer inside a cell, that is put in the groove, and the T cell says, I've never seen that before. This is foreign also. So now you have T cells, both the helper and cytotoxic, being involved in fighting cancer, which is an awesome thing. And there's something called immune surveillance, meaning, believe it or not, we all probably have cancer arising once a year, once a month, and your T cells are using immune surveillance, using the MHC1 and MHC2 receptors to say, hey, this cell probably needs to be destroyed, it's cancerous. That's going on too in the background with MHC1 and MHC2. Questions or thoughts? Moving fast.
Okay, yeah. So, Paul, are you following all this?
I'm trying to. I'm a geologist, so I'm kind of the proxy for the audience here because I know nothing about this subject. So,
yeah.
But this is fascinating. To me, it's just so phenomenally complex, and yeah.
Well, yeah. All right, so let me ask a few questions on behalf of the audience then.
Sure. Yeah.
You use a word and you've used it multiple times now, and I think we probably need to define that. And that word is receptor. So
right.
What is a receptor and what's it doing?
Yeah,
as a general thing.
That's a good question. A receptor, pretty much all receptors we know are proteins. And when we talk about receptors on the outside of a cell, those are proteins that receive messages pretty much all the time, or sometimes when they're needed. So we have growth factor receptors. And all your tissues have growth factor receptors on the cells. And when that tissue is not growing like it should, it puts a few more receptors out there and it says, bring some growth factor over, and it grows the cell. So receptors are very important for a lot of reasons. One is cell growth.
Yeah.
And interestingly enough, in breast cancer, that's what causes breast cancer. There's all of a sudden too many growth factor receptors appear. And that causes one form of breast cancer that we're curing today by shutting down that receptor with an antibody. So receptors, an antibody is a soluble receptor. An antibody is this beautiful protein that has two prongs on it, and it binds anything and if it's made by this system to wake up and tell the B cell to make antibodies, it's made to specifically bind now to that antigen. So everything I'm talking about here is a receptor. The antibody is a receptor. The phagocytes have receptors on the surface for bacteria every day. So very important. When I read go ahead.
Yeah, I was going to say I like when I'm teaching cell biology, I like to tell the students receptors are like the eyes and ears and nose of the cell, right? The cell is bounded by a cell membrane, but it needs to be able to interact with the environment that it's in, right?
Yes.
Your cells need to know, for example, my nose the goblet cells in my nose. They need to know, there's something in here. I gotta make mucus and blow it out. That's something that a cell does, and those particular cells are called goblet cells, right? So
Right.
it needs to be able to recognize naughty things trying to go up my nose, and
Yeah.
so and that's what a receptor is all about. It's a really cool molecule because in biochemistry we call it transmembrane, right? So you got your cell membrane, and the receptor goes across that membrane. There's a part that's outside, and there's a part that's inside, and the outside part is the part that's the eyeball that's looking around to see what's going on, and maybe it binds to something and says, this is weird, and that transmits a signal through the membrane to the part on the inside, which tells the cell, hey, there's something going on out here. You better wake up and do something. And so, yeah, that's kind of what a receptor is. And yeah, you're right. Receptors are absolutely important because they're the things trying to figure out what's not right here in the body. You don't want an autoimmune disease where your immune system starts attacking yourself, right? But you need to be able to distinguish what the bad guys are. Anyway.
Exactly. That's a good
Receptors.
point. I'm glad you backed up on that because we glossed over that. Yeah, we're talking about receptors here. MHC1, MHC2, the T cell receptor, all important. They're talking to each other through those receptors.
Absolutely.
And I think another thing to add, when I taught cell biology, is you can have thousands of these they're mostly protein receptors. What's interesting, though, that could get out of control. You could accidentally engage receptors. So what I think is fascinating in cell biology is there's more carbohydrate on the surface. And your immune system kind of ignores carbohydrate because it's not as complex as proteins. And so there's a beautiful design here. Thousands of protein receptors, but you have probably millions of carbohydrates. And so I tell my students, I had a great student years ago. She called everybody sugar, her teacher sugar. And so the cells, yeah, we're talking about an amazing complex surface with these receptors that can communicate. But I'm working on a theory that these carbohydrates are probably keeping that muted somewhat. So your cells look over there and say, oh, there's my friend. Yeah, he's got the right carbohydrates, or we're good. He's covering up his receptors. And so the cell surface is fascinating. That's our point.
Yeah.
Here.
Oh, yeah. Yeah.
And the MHC works in that environment. Here's where we can, so now we've got the basics of the immune system: recognize a foreign invader, call out the troops, recognition and response. The MHC plays an important role in self-preservation, in saying, this is me, don't attack, or this is not me, you're going to probably need to attack. So, but here's where it gets complicated. The MHC2 was not named because it was an immune receptor. It was named because of transplantation biology. So major histocompatibility complex is a set of receptors that we all have. And if we're not matched, having the same MHC, we can reject a transplant from each other. That's how it's discovered.
Yeah.
So histo means tissue.
Yeah.
Major tissue compatibilities. It's discovered. So wait a minute, that's a separate subject from the immune system here, or is it? Because and then why do we differ so much at that level? So right now, if Todd needed a kidney, there's a high percentage that Paul and I would not be able to donate. To find a donor, let's say in the American population with 300 million people, it's like 1 in a million to find those MHC matches. And each MHC receptor, MHC2, has 2 proteins. One can come from your mother and one can come from your father, or they both could come from your father and both from your mother. So that multiplies the number of MHC you have on your surface. So if you want to get a kidney from your child, you have a 25% chance of getting that finding the right child for that. because the mother's proteins are in there. So these are codominant, what's in genetics called codominant receptors, meaning they're made from two pieces. One piece can come from the father, one piece could come from the mother, and so on. So that multiplies the number of MHC. So the question is, why did God make us differ that way? And I was going over verses this morning. If you look at some of those verses where David says, "You made me this way," that's very personable. It's talking about David. I knew you and your inward parts. Yeah. And I know I want you to do this in the church and you to do that in the church. Remember the twins, Isaac and Esau, different, hairy and non-hairy and different, unique. There's something there that's so beautiful. You know, we just had 4 grandkids over there yesterday. We have 8. I can't believe how different they are. And they're from the same family. Why are they, this one is crazy over here, this one's nuts over, they're with that age. and this one wants to play with this. I had one yesterday just want to look at birds. That's all he wanted to do. So there's beauty in that uniqueness, but then this transplantation thing is beautiful too, because we can be cured. So I tell my students, wait a minute, 2 years ago, our neighborhood, we had people breaking in. And sheriffs came to our house and said, you need this kind of lock and this kind of lock, and we locked things down. And can you imagine the market of organs if we all could trade our, could you imagine the crime involved in that? Yeah. I won't name the country, but there's a country in the world where they have found ambulances going away from the hospital and people coming up and buying organs. That happens in our cultures. I'm thinking, God knew how bad we were going to be. Yeah. He knew, hey, I like that guy's arm over there. I'll just go over tonight and take that. So I tell my students, he knows us. He knows how defiant and evil we can be. So that's kind of one explanation, plus just the uniqueness explanation. But here's where it gets, here's some data to actually support that in biology. And it gets, now it's going to get really interesting and really strange. We have mice, okay? I started out at Michigan State. In order to pay my way, I worked in a microbiology lab at night. I love sitting in the offices and reading the science magazine and acting like I was a researcher. And then they said, you got to clean the mice cages and you got to watch over them. And I'm going, these are fascinating creatures, but they smell. Believe it or not, mice can, using the MHC, smell another mouse, and they will choose a mate who has a different MHC set of receptors than theirs.
What?
Yeah, they will choose that mate because he smells different, and we now know, they've tested it to show the MHCs involved in odor detection. You say, wait, why would they want a spouse with a different MHC? Well, if you just keep marrying a spouse mouse, and that's a new one, and mouse they're the same MHC, your children are going to have less variable MHC and less ability to fight off infection. So here's God's plan saying, yeah, I'm going to throw all this, you guys deserve this mess of microbes, but I'm going to show you my mercy here in allowing this MHC to help protect you from massive episodes, like the COVID epidemic. There's evidence that we were protected because of the MHC, and God saying, I want humanity to make it till Christ returns. I think of it that way. And so you have, and here's another one. So in char fish, okay, now you got to go to the Arctic Circle. You got to really like ice fishing to go to the Arctic Circle.
Yeah, you got to really like it.
And you can go, I wish I could show you this image. There's a guy with like an actual fishing pole instead of a ice fishing pole. And he's got this beautiful red, white spotted fish. It's a char. Well, we now know somebody took these char, they're really easy to grow in the lab, and he took water from one char family and that had a different MHC from the other char family, and the babies wouldn't swim in it. The babies would only choose the water of their own MHC. Well, that kind of makes sense because the babies don't want, they know that's their family. So now you have baby char choosing their own MHC because that's their brother and sister. And maybe the non-brother and sister could be more of a predator type because we know that fish will eat fish. This gets really. And then they say in some of those, they've tried different fish now in the lab, and they say the adult fish will lose the ability to detect their own MHC, and they will marry another fish with a different MHC when they're adults. So something's going on. Something's going on. God is saying, I want to preserve diversity.
Yeah.
And
That's fascinating. I
Yeah.
I was going to ask, we've talked about humans, we've talked about mice, we've talked about fish. How widespread are MHCs? I mean, do we find these in all vertebrates?
All
basically?
vertebrates so far.
Yeah.
So lizards,
Yeah.
they've had lizards choosing mates on odor. Birds, they can't figure it out. There's a guy who did a bird study because I guess birds don't smell much very well. And they found by taking the bird, the husband and wife birds and looking at their MHC, they were choosing different MHCs. They don't know. They don't know how the birds. They said, could it be plumage? A weird plumage they're looking at, or okay, this gets PG-13 rated. We've had experiments on humans.
I was going to say.
Okay, we'll keep it. We've got to keep it family. Okay, how do we say this? They took T-shirts from boys, and they let girls smell the T-shirts, and they chose alternate MHC. I don't know if we're going to edit this or take this out.
I suggest that we not share this with our wives because I don't want them to think that it was biological determinism, honey. It wasn't anything about love or any of that stuff.
We have a limited audience for this podcast today. It's odd, and the people who write this up, we say the data is not that great, but it keeps repeating.
Yeah.
Because
there's our aftershave manufacturers kind of missing a trick here, is it?
They could be. But leave it to God to use a sensory. What sensory system is he going to pick to preserve MHC differences? He wants to preserve MH. It seems like that's the design feature here. In this fallen world. I had a friend during COVID. I don't want to get on COVID, but Christian friend, colleague, I won't say he just said, I listened to this guy who says we're all going to die. And I'm going, has the millennial kingdom come? Has Christ come?
Right.
And we think we're all going to die. But it seems to me that God wants us to make it to the point when Christ returns. And I think he's doing everything he can so we don't kill each other and just absolutely wipe each other out. And he's using the MHC here to say, I don't want a pandemic to wipe you out. I'm going to show you why. It's kind of cool to think about that. So
yeah,
interesting.
Yeah. That's remarkable. Yeah. Okay. Yeah. Definitely don't tell that to my wife.
Yeah. It's lizards, birds, several fish species, and mice. And I don't know if I can do the mouse experiments because I didn't like the cleaning out the mice. Maybe I don't like any of their MHCs. It was really bad. Oh.
All right. Well, okay. So that is a remarkable system, the MHC system here. And yeah, it does make sense because if you put, if your MHC is monitoring self versus something else, and you put the wrong organ in someone, it makes perfect sense that it would go into organ failure because the immune system is going, this isn't us. This isn't us.
Yes.
Yeah,
Right.
so
Right.
it gets
Right.
attacked.
Also,
And
you
just
have
to be clear, we
Yeah.
didn't discover that with human transplants, right? We'd worked that out before we started with that stuff, right?
I think so. Yeah.
Yeah.
It was with mice, I think, wasn't it?
Yeah,
The
and
mouse
it would have
model.
been a number of different types
Yeah.
of animal
Mice
models.
are really heroes. I didn't like having to cull a whole group when they got older, all that kind of stuff, but they have saved the day in our learning of immunology, and they're still used today, all to make antibodies. But now we're actually making antibodies in plants, and so we can spare some mice
What?
So
How are we? Are these transgenic? Are these genetically engineered plants that make
yeah.
antibodies for us?
So if you think about it, one of the problems with mouse antibodies is they have their own set of pathogens that could contaminate whatever we make in a mouse.
Sure.
That could cross over. So what they're doing now is taking human antibody genes, putting them in mice, and then taking that system out and putting it, and then having the mice regenerate some DNA that we can get it, then cleaning up that DNA and putting it in tobacco plants. And now we have tobacco plants that don't have our viruses, don't have human or mammalian viruses hanging around.
Yeah.
And
sure.
so we just chop up them. So a few years ago in the Samaritan's Purse Hospital, when Ebola, you might have seen this on TV, the Dr. Brantwell that came home, he survived Ebola because we got a monoclonal antibody produced in tobacco plants. Franklin Graham got this done. That one dose, there's only one dose in the world, and it went by airplane, canoe, hikers to get it to those missionaries, and they started feeling better when the 2 doses were given to them, and it was tobacco plants that saved the day on that one.
Yeah.
So yeah, we have tobacco plants making
amazing.
antibodies.
And just to be clear here, nobody's smoking these antibodies. They're not
Yeah,
chewing the
I never
antibodies.
thought about that.
You're grinding up you're just using the plant to make it, and then you grind up the plant and get the
Right.
antibody out.
Yeah.
That's right.
Right.
Okay.
Yeah.
Yeah. Okay.
Yeah, I'm
Good.
too much of a strict Baptist. No smoking, no drinking.
No
so
smoking.
I
Yeah.
thought
No
of
smoking.
that.
Well, you're in Southern California. You definitely shouldn't be smoking.
Yeah, that's right. I
That's
never
for
thought
sure.
about. Yeah, maybe they could smoke it rather than inject it. Oh boy, I don't know. Yeah, so those receptors, all those receptors, the antibody receptors, the white blood cell first responders, it's receptors. It's the feeling and touching with a receptor, and the MHC. And so this is what makes the MHC so interesting. It's more promiscuous. It's more that the peptides in the groove don't have to be perfect to make the system work. So it's really, you have some receptors that are really tuned in on one thing and they got to go get it. But the MHC is kind of wide open to making more. The end result is making more antibodies, making a whole bunch of different antibodies. So that groove, it's interesting. That groove where they bring up the peptide, it's only 8 amino acids long, 8 amino acids. And it brings up different ones each time. And so the T cell is going, oh, we better make antibodies to that one, and we better make antibodies to that one. And it's not covalently bonded. That little peptide just kind of floating in there. So the T cell's going, oh, we need to make antibodies to that and that and that. So it's a beautiful system because it allows for diversity of antibody responses. Whereas if the T cells were just in charge, they make something very specific, and you wouldn't have diversity of antibody. Yeah. So that's another part of the MHC that's beautiful.
Yeah. And let me clarify that for our audience again.
Yes.
As a biochemist, 8 amino acids is very, very short.
Yes.
It's so your typical, and I'm going to say this for bacteria because that's the system I know. Your typical bacterial protein is made of about 300 amino acids. So 8 amino acids is really small. It's actually quite a bit smaller even than insulin. And insulin is just a little tiny thing that is used to regulate your sugar. And that's something you've probably heard of, insulin. But that's a really small protein as well. And so this eight amino acids, it makes me scratch my head and think, is that enough to be specific? But apparently, that's enough to be specific.
Yes. Yes. Yeah. And here's where it gets more interesting. T cells play an important role here. Remember, they're responding. So when you're a fetus, these T cells are being made, and they're making every receptor we can imagine to all of those peptides. Say, wait a minute, that means you're going to react with self. Well, at the fetus stage to birth, there's a school that takes place, and T cells are schooled in what they can do. And if they have a self peptide, they don't just fail the test; they're killed. This is a nasty school. This is a bad school. You don't want to be a student in that school. But so get it. The T cells actually have to be educated to know what self is versus non-self, and a lot of them are eliminated that react with self. Because if that MHC brings up self, you don't want a T cell saying, "Oh yeah, kill it." So they have to be schooled when you're a baby or pre-baby stage. That's going on too. So, and where does that information come from, and how is that wired in through our families? So amazing stuff.
All right.
So back to natural. Go ahead. Yes.
Yeah, I was going to say, watching the clock here, making sure we got everything in here. Our theme here is natural evil. So we've been talking about the immune system and the major histocompatibility complex, aka MHC. For those of you who don't want to stumble over major histocompatibility complex, can you kind of give us some ideas? Now, you've already mentioned how this is a great example of God's grace, and I think that's remarkable. Yes, God has set it up so that we are not going to be constantly taken down by unseen bacteria and viruses in the environment. And he also set it up so that we are not going to easily butcher each other for organs that we need. Thou shalt not covet thy neighbor's liver or lungs or whatever. So that's probably good, because you're right. That's exactly what would happen. But what about if the world hadn't become corrupted by sin? What do you think the prospects are for understanding the immune system, this recognition system?
Yeah.
What could it be doing if there aren't really a lot of these harmful bacteria in the environment?
This is the hard question.
It is. That's why I asked you.
Well, let me tell you a story, and this is where Todd can help us here on this idea. When you're a scientist at church, or someone will say Dr. Francis, which I don't want to be known as church's Dr. Francis, they'll come up and say, I got this leg problem, and I'm a PhD and I'm not an MD.
Right. Yes.
And they'll bring you books. And so about 10 years ago, lovely lady in the church, she brought up a book on diet by blood type. So we are all separated from each other into 4 or 5 groups of blood type. We can't transfuse unless we have the same blood type. And it was called Eating by Blood Type. And I started looking at it and I'm going, here's the Tower of Babel. God's going to separate people by language. Could he have been separating people by food choice? I'm going down the road with my family, and it's always like, where do we go now? McDonald's or this one? Can someone make a decision? No, I like this. I don't like this one. I don't, I'm just wondering if diet by blood type has a little bit of truth to it. And so maybe the MHC. So if we look, if right now in the United States, if we can only find a donor out of 1 in a million, that means there's 300 different people groups separated by MHC here. That's interesting. How about Neanderthals and others leaving? Could some of this incompatibility idea be part of their migration? And distance from each other? May smell. You guys smell different than I'm going this way.
Yeah, I bet they did smell.
So I think that's a whole area to be possibly looked at. Separation of populations. So God is saying, I got you separated by different populations, and maybe we need to take a look at that at the genetic cell surface level.
Well, he did command us to be fruitful, multiply, and fill the earth. So,
Yes,
um,
yes,
and he doesn't want us just sitting in one place. He wants us to take advantage of the fullness of the earth that he's given us. So it does
right.
make sense that would be that he would build in preferences, things like that that would help us to differentiate
Yeah,
from
and
each other.
when we travel, we can get food illness in a different culture, sometimes not from a bug, sometimes just from the food, maybe.
Yeah.
And so that was another proof that God was, I want you to go over here and make your own food and eat it. And that ends up being beautiful to us, though, because now I'm saying, I'm not Asian, but I love Asian. There's that part of it too. That
Yeah.
makes our cultures unique and beautiful. At the same time, does this all go back to how he used that to partition us
Yeah.
into groups? I don't know. I think
What
it's
about
neat.
what about microbial recognition? Now, we've had you on here before and we've talked a little bit about this. Do you think in the original creation, is there a place for recognizing bacteria?
Yeah, because we now know the microbiome is real. It's part of our health. This
Mm-hmm.
kind of goes back to a talk I did at the ICC, and I've struggled. And one of our retreats, I talked about this. I struggle with. So I teach microbiology.
Mm-hmm.
On the same day, I would teach immunology, and I go in and talk about microbiology. We talk about, look at this evil, protein right here. It can cause death within a few days if you get sepsis. This gram-negative, it's a cell wall component. If we inject into someone, they're going to get the same problem and die unless we neutralize it. And then I go to the microbiome, and the E. coli that cause disease, all the E. coli have that same protein. So why are we dying by the minute? Well, the microbiome really kind of sits on all our outside surfaces. Your gut is really an outside surface.
Mm-hmm.
And I started thinking about this, and our immune system actually talks across that barrier and says, okay, I see E. coli's there. That's good that you stay there. You're providing vitamins on that side of the mucus. You're not going into the tissues. That's good. We're all happy here. So it looks like the immune system, to me, we could possibly argue was built to maintain that important microbiome tissue,
Right.
to do amazing things.
Right.
And you could say, isn't that bad design to allow it to get in the wrong place? Well, no, because I don't think of my car air filter being bad design. I think it's essential to not let dirt into my engine. We live in an interactive world. God made oxygen that can burn down our forests, but we breathe it too because we have to interact with something to breathe. So that's some other thinking I'm doing is could our immune system have been built as is to make us unique, but also to maintain that microbiome? Lots of curious things there.
Yeah. Yeah, I've heard
I
you
mean,
use.
this may be a naive comment from someone who doesn't know anything about these topics, but is there an analogy here with, say, human skin? Skin is a protective barrier, right? But it's going to be just as useful in a pre-fall world as in a fallen world in terms of kind of
True.
keeping certain things in and keeping certain things out.
That's what I'm saying. Yeah, absolutely.
Yeah, so it's the immune system could be seen in a similar way. It's kind of keeping things where they're meant to be, right?
Right. Right. Yeah, and we have more and more about this gut brain axis where they've transplanted microbes from long distance runners into mice, and those mice will run longer on their treadmills. Something's going on. That microbiome may have more to do with our brain than we think, but our immune system has to keep it in the right place. Just like you don't want your kidney in your throat, you just got to keep it in the
Yeah.
right place.
And that's part of
So,
the good design, right? I mean,
yeah,
design is not just everything is happy all the time. Design is everything has its place in creation
yes,
and
yeah,
a job to do. and you don't want things wandering off. You want to be able to find the right guys and recruit them and get them into the right places. But you don't want things wandering off where they're not supposed to be. And in the realm of microbes, I mean, they don't know. They don't have brains. You can't teach them this is where you're supposed to be and this is your job that you're supposed to do. They're just little microbe machines. So it makes sense then that you would need a system that would be able to regulate or to help to regulate the microbes.
Right.
Now you just gave a little story there. We're running out of time, but I gotta hear more on this. Transplanting microbes from runners to mice. Exactly what do you mean by that? I mean, are they what is that?
Yeah, it's a fecal transplant, which
oh, lovely.
yeah, sounds. I mean, but at least they're not going inside our body and grabbing something. You just have to send a sample in. But
Yeah.
There's this gut-brain axis. They've taken microbes out of children with certain mice with lactose intolerance. Children from children's diapers, put it in mice. They get lactose intolerance where they never had it before. That can be explained in a lot of different ways, but there's more. There's some other behaviors that are coming to my mind right now where they've transplanted to mice, and mice have picked up that attribute that humans have. It's pretty freaky.
Yeah.
And part of it is we're more like mice than we think, maybe.
Yeah, well, there's that. And yes, folks listening, fecal transplant is a thing.
It is a thing, yeah. Yeah.
And it is used therapeutically because your gut is designed to work with the bacteria that live there.
Yes.
And if your bacterial population gets out of whack for a long time, you need an infusion. Basically, you need a transfusion.
Yes. Yeah.
Now, they don't cut you open and they don't make you eat nasty things, but there are non-invasive ways of transplanting basically microbial stuff from a stool sample into someone else's gut. And you know what? It helps them. So
right. And we don't go too far off kilter here, but I just listened to a fascinating podcast about our microbiome whereby some E. coli were making tangled fibers, and because of the gut-brain axis, those tangled fibers were contributing to Parkinson's in the brain.
oh my goodness.
So it's not all good. We got some bad going on there.
Right.
But that's where the fecal transplant could be used to push out the bad, overgrow the bad.
Yes.
This is really getting interesting. Now, the researcher, oh man, he had grants from everywhere, but he finally said, this only explains 20% of Parkinson's. But man, wouldn't it be great if we could figure out the other 80%, if there's more microbes in being involved.
Yeah.
But that's amazing. Tangled fibers from microbes. So it looks like that axis is really going on. It's really happening.
Yeah.
Because you have this blood-brain barrier that you don't want anything getting through. So my question to him is, how does that tangled fiber get through? I don't understand that part.
Yeah.
Just to tell you, but he's got the evidence. So he was using mice, these poor mice. He had them walk on a plank, and when they had the bad E. coli, they would fall off the plank. I mean, they had the Parkinson's syndrome. He showed it.
Wow.
So pretty wild.
Yeah, I think we're only just beginning now here in the 21st century to really appreciate and fully understand how valuable and important our bacteria are to us.
Yes.
That we could not be healthy adult humans or healthy, we couldn't go through the proper growth if we didn't have the right bacteria. And the idea that we should just blitz them all with antibiotics, that's no good at all. That's really bad. And I think I'm wondering, over the next 100 years, what are we going to find? What's been going wrong because we've been doing that kind of thing, using our antiseptic cleaning agents and our antibacterial cleaning agents, and constantly washing our hands with soap and water. That's all good,
Yeah.
but we also need good bacteria to keep us healthy and to keep us safe.
Absolutely. Yeah, I mean, we've all had strep throat, and we benefited from the antibiotic that we need.
True.
But could that antibiotic come with a culture of good bacteria to take its place, or drug resistant in a certain way? And you got to be careful with that. You don't want to make too many drug resistant ones, then it could go bad. But
Yeah.
I'm sure there's some exciting ideas out there on that.
Yeah.
So,
All right. Well, the MHC is, let's see if we can recap what we've learned here. The MHC is all about, it's a receptor. It's a little molecule that sits on the surface of your cell, and it recognizes stuff. It presents stuff that's going on to other cells in your immune system, and your immune system can recruit cells to kill things that are making the wrong stuff. Your cells can, your immune cells can recruit other immune cells to spread the word. They can start making antibodies, which helps to spread the word as well. And it's all sort of mediated there through this major histocompatibility complex. And it shows God's grace, because it is by God's grace that we are not consumed, right? That's what it says in Lamentations there, Lamentations 3. And so, we are constantly surrounded by these bacteria, and they're always looking for a way to sneak inside and take advantage of our nice, warm, wet interior. And they would love that to multiply in, and our MHC and our immune system keeps that from happening. So that's a good grace, a good use of things. And then in the pre-fall world, we think, there's a number of different functions that it could also perform, but one of those might be just making sure that bacteria stay where they're supposed to be, and viruses as well, and making sure that the good guys are doing their good work. So, all right, any final words that you wanna give us to wrap this up?
Yeah, I'm gonna give one caveat that's impressed me lately, and that is we have good bacteria. And you guys covered viruses on a podcast, which is very good. But just to reiterate, we don't have any good human viruses or good animal viruses that stay on us to pretty much do anything good. We have good bacteria. We find E. coli in cattle. We find E. coli in us. But it's kind of interesting that our immune system wipes out all the animal viruses except two. There's a JC virus and another one. But they can also cause brain. So why is that? Why does our immune system say, I'm going to tolerate these bacteria, but not viruses? There's something there. There's something important there. I can't figure it quite out, but that's why we need some more creationists, some young creationists to work on these things.
That's right. Young creationists out there, listen up. Yeah, we definitely need that. And you're right. Viruses are just really weird, and they look like killers, and so that's one of those.
My final thing is wash your t-shirts so you don't attract the wrong mate.
There you go.
The wrong wife or husband, I should say.
That's right. That's right. All right. Well, thank you very much. I think we've neared the end of our episode here. As usual, we don't know what's coming next. Actually, it's kind of a surprise because we actually don't have any episodes pre-recorded right now other than this one. So we literally don't know what's coming next, but I'm sure it'll be great. I'm sure it'll be a fun conversation. Paul, you got any closing thoughts here?
No, I just want to thank Joe. It's always fascinating to listen to Joe talk about these topics. So we're really grateful for you coming on again, Joe. It's been great. Yeah.
Well, thank you. Thank you both. And I'll definitely advertise it to my family, maybe PG-13 rating, but we'll see. But no, my family's been blessed. And I was in a meeting at Masters 5 years ago, and we were rotating some homeschool groups through, and on the way out, two women said, "Have you listened to this podcast?" And it was your podcast, and I'm going, "Wow, it's getting out there."
Wow, that is great.
That's good to hear.
Oh, that is very good to hear.
I know that guy.
Very encouraging.
No, you don't. I said, "Yeah, I know that." All right.
That's
Well,
great.
Thank you,
great.
Bob.
All right. Well, thanks very much.
Yeah.
We will catch up with you again, audience members, in 2 weeks. Don't forget to like and subscribe and hit that bell for notifications and all that good stuff. And remember, we are a listener-supported ministry, and we do need your donations to keep this all going. So please visit letstalkcreation.org to find out how you can make a contribution, and we will see you next time.
See you then.
Thanks for listening to this episode of Let's Talk Creation. For more information, visit us at letstalkcreation.org, where you'll find an archive of past episodes and all our show notes. If you'd like to leave a comment or make a suggestion, you can find us on all the major social media platforms. Let's Talk Creation is brought to you in the U.S. by Core Academy of Science and in the U.K. by Biblical Creation Trust. As a listener-supported ministry, we are grateful for all of your financial support. Find out how you can make a contribution at our website, letstalkcreation.org. Also remember to like, subscribe, and share this episode with your friends. Thanks, and see you next time.